I'm pleased to have Dr. Maciej Czerwinski here with us today. Maciej received his PhD from the University of Maryland at Baltimore Medical School in the Department of Pathology. He has been with XenoTech since 1999 and currently serves as a Director of Consulting. Maciej guides the development of products and services for the analysis of drug metabolism and protein expression. Maciej also conducts research on drug-drug interactions between biologics and small molecule drugs, and is the inventor of XenoTech's single-freeze enhanced hepatocyte product, CryoStax. It is my pleasure to present to you Dr. Maciej Czerwinski.
Good afternoon, everybody. Thank you for attending our webinar. Today, I'll describe XenoTech approach to evaluate in vitro drug interactions of immunomodulators. Aim of our presentation is to inform you about our approach to evaluate potential drug candidates to modulate expression of drug metabolizing enzymes, with emphasis on immunomodulators, which exert their effects indirectly through action of cytokines. Building blocks of our presentations are definition of immunomodulators, background on immunomodulator drug-drug interactions, regulatory guidance in this area, XenoTech study design to address the issue, and 3 case studies done according to our experimental design. Most broadly, immunomodulators are drugs that adjust immune response. Structurally, they may be large or small molecules. New molecular entities of large molecules appear frequently in this arena. For our presentations, immunomodulators are compounds that are able to stimulate cytokine release or change cytokine levels or activity. Our assay, in its current format, is suitable for some, but not all, immunomodulators. Our emphasis is on compounds affecting cytokines, more specifically drugs that stimulate the release of the cytokines in whole blood. It is anticipated, although not specifically demonstrated, that the effects of cytokines released from Kupffer cells are also accounted for in our experimental design. The following sequence of events illustrate immunomodulators perpetrating drug interactions. It is known that factors such as viral and bacterial infections, vaccinations, inflammatory diseases such as rheumatoid arthritis, chronic diseases, and certain therapeutic proteins result in patients in fever, changes in the levels of pro-inflammatory cytokines, exemplified here by interleukin-1, 6, or tumor necrotic factor alpha, and changes in acute phase reaction proteins. Positive ones here, exemplified by C-reactive protein, alpha-1 acid glycoprotein, and the complement factors. Changes in these regulatory molecules can lead to suppression of drug metabolizing enzymes, such as CYP enzymes. Subsequently, increasing systemic exposure to hepatically cleared drugs, which may lead to drug toxicity and/or exaggerated pharmacology. I would like to emphasize that therapeutic proteins may change not only levels of so-called pro-inflammatory cytokines, but so-called anti-inflammatory cytokines as well. Analogously, acute phase reaction proteins are those that are so-called positive, as listed on the slide, and they are increasing inflammation. And there are also groups known as a negative acute phase reaction proteins, and they are decreased in inflammation. And examples of those include albumin, transferrin, or retinoid binding protein, for example. Immunomodulator drug interactions, or the ability of vaccinations, cytokines such as interferon, interleukins, to cause clinically significant impairment of CYP-dependent metabolism of drugs has been known for many years. Some cytokines suppress CYP enzymes by acting directly on the hepatocytes. Examples are interleukin-1, 6, or tumor necrotic factor alpha, but not others, such as interleukin-2. Others suppress CYP levels indirectly by first causing the release of cytokines from Kupffer cells or peripheral blood mononuclear cells. Examples of those are endotoxin, which we use as a positive control in our assay, and synthetic and bacterial oligos that contain pathogen-associated molecular patterns, such as CpG motifs. Cytokines can suppress CYP levels by activating certain transcription factors, such as NF-kappa beta, STAT1, CAT enhancer binding protein beta, and others, which diminish the transcriptional activity of other nuclear receptors, such as CAR, PXR, glucocorticoreceptor, and hepatic nuclear factors. Diminished drug metabolism in inflammation may have more than one underlying mechanism. We recognize the effects of increased levels of pro-inflammatory cytokines, leading to changes in transcription factors, reduced transcription of CYP genes, leading to lower amount or lower abundance of CYP proteins. But pro-inflammatory cytokines can also induce nitric oxide synthase. Cytokine-inducible nitric oxide synthase, INOS, produces large quantities of nitric oxide upon stimulation, such as by pro-inflammatory cytokines, for example, interleukin-1, or tumor necrosis factor, or interferon gamma. Intrahepatic production of nitric oxide can lead to killing the invading microorganisms, suppression of CYP enzymes, or death of hepatocytes themselves. To keep up our regulatory awareness in this area, we note that 2012 FDA guidance referred to therapeutic proteins as potential perpetrators of drug-drug interactions. Although this subject was not covered in 2017 guidance, 2012 guidance noted low potential for small molecule drugs to affect therapeutic proteins, with some exceptions, and focused on therapeutic proteins such as cytokines or peptide hormones that are cytokine modulators causing CYP suppression. In that guidance, an example of desuppression was also covered. Desuppression being a return of pro-inflammatory cytokines to pre-disease level and return of suppressed enzymes to pre-disease level as well. This document noted that in vitro or animal studies have limited value in the projection of clinical interactions, and therefore clinical cocktail studies were suggested. I would like to add that this comment about limited value of in vitro studies referred to state-of-the-art at that time, which is not necessarily the case today. At that time, application of therapeutic protein directly to cultured hepatocytes may have been considered state-of-the-art. Other scenarios mentioned in 2012 FDA guidance are those in which therapeutic proteins are used in a combination therapy, in which case evaluation of the effects of each product or another was suggested. Another scenario was when there are known mechanisms or prior experience with certain PD or PK interactions, appropriate in vitro or in vivo assessment should be conducted. In Europe, EMA 2013 guideline on the investigation of drug interactions specifically stated that interactions with therapeutic proteins, including peptides, are not discussed in that guideline. And reference was made to 2007 document. EMA 2007 guideline on clinical PK of therapeutic proteins stated that some therapeutic proteins, for example, immunomodulators such as cytokines, have shown a potential for inhibition or induction of CYP enzymes, and thus the need for in vitro or in vivo studies should be considered on a case-by-case basis. Again, in 2007, it was stated that we lack knowledge about suitable tools to explore such interactions. Certainly, that was a document earlier than 2012 guidance from FDA, and it's understandable that the state of the art at that time was different than it is today. In Japan, PMDA 2017 drug interaction guideline contains language or recommendations very similar to that of the FDA documents. I'd like to add that all these guidance documents are available from our website for your further readings. Is new FDA guidance on DDI evaluation for therapeutic protein coming soon? Well, we certainly expect so. FDA posted a request for information and comments in May of 2018. These comments and the responses from the industry received by FDA are known as a FDA docket, which is a reference here. And if you receive the copy of the slides, that would be a live link to FDA docket. In that request for information, FDA asked, in what scenarios and for which classes of therapeutic proteins should DDI assessment be performed? Second, what types of assessment can be useful? And thirdly, what are the study design considerations? Comments were received from pharma companies, 8 of them responded, the IQ consortium leadership groups, 2 of them, and 1 CRO, XenoTech. Many recommendations were given, including risk-based assessment that would take into consideration specific patient populations, recommendation that FDA should state their position on the utility of in vitro assessments for therapeutic protein DDI prediction, recommendation to restrict recommendation to therapeutic proteins that modulate pro-inflammatory cytokines. We certainly agree with that. Recommendation to allow population PK analysis of DDIs, and recommendation that in vitro studies with the therapeutic proteins should not be required when clinical translation had been previously established. XenoTech also provided our comments to FDA request for information. We stated that in vitro testing in hepatocytes typically has been limited by 2 factors. First, investigating a single recombinant cytokine at a time, while in vivo we deal with a plurality of cytokines. Secondly, absence of other relevant cell types in the test system, and we certainly recognize that peripheral blood mononuclear cells are a major contributor of cytokines that reach hepatocytes. XenoTech has developed in vitro system to evaluate xenobiotics as immunomodulators of drug transport and metabolism in human hepatocytes. This experimental system is based on a treatment of whole blood by therapeutic protein, followed by treatment of hepatocytes with the plasma that allows for indirect effects of the therapeutic protein to be evaluated. We also know that plasma from patients with certain diseases with the inflammatory component can be used in our studies. We hold certain IP position in the US patents listed here, as well as other applications. Schematically, in our study design, steps to evaluate direct effects are listed here on the left-hand side, and steps to evaluate indirect or cytokine-mediated effects are listed in the right column. Analysis of direct effects is based on treatment of co-culture with the drug and is followed by measuring mRNA and enzyme activity in a way that is analogous to design of our enzyme induction study. The first step of analysis for indirect effects is treatment of whole blood with the drug. We prefer to use fresh blood to avoid any cytokine release that is associated with processing blood or trying to isolate PBMCs from there. We typically quantitate cytokine with mesoscale discovery assay, although other platforms are suitable for that purpose too. Subsequently, we treat co-cultures with the plasma stimulated by therapeutic proteins, and it follows with measurement of the mRNA and activity levels, the same way as it's done for evaluation of direct effects. I'd like to now go to our first case study, that of anti-CD28 antibody. Initial proof of principle study was done with anti-CD28 monoclonal antibody. You may recall that the infamous antibody TGN-1412 was also directed against CD28. That antibody caused severe morbidity in a first-in-man study in 2006 in London. 6 volunteers became sick as a result of the cytokine storm. This trial initiated extensive examination of steps that were in place to assure safety of the volunteers, but failed at that time. We wanted to evaluate the TGN-1412 antibody itself, but it was not available to us. Other superagonistic antibody against CD28 was available. It is called ANC28. CD28 is one of the T cell receptors. Stimulation of T cells could be beneficial for the anti-cancer effects. TGN-1412 antibody was an agonist of CD28, mimicking action of antigen-presenting cells, receptors CD80, antigen-presenting cells, the receptors CD80 and 86. When antigen-presenting cells are not adequately stimulated by neoplastic cells, tumors may evade detection and destruction by the weakened immune system. You can imagine that application of antibody engaging CD28 receptors on T cells may alleviate the situation when this engagement is incomplete. Incomplete engagement results in a T cell apoptosis and anergy. Substituting CD80 or 86 with the anti-CD28 antibody may result in a proliferation, differentiation, and effector functions of the T cells. As you recall from the earlier slides, first element of our study design is treatment of the whole blood with a test article. For this study, blood was collected from 4 donors and treated with 1 concentration of the antibody for 24 hours at 37 degrees in an atmosphere of normal air. We evaluated 9 cytokines in the collected plasma, and plasma was given to hepatocytes co-cultures from 3 donors at 10, 20, or 50%, followed by analysis of mRNA and activity of free CYP enzymes. Following stimulation of blood, cytokines were measured. This table compares levels of cytokine release in plasma treated with saline, our negative control, LPS, our positive control, MAPC antibody, which is an isotype control antibody, and anti-CD28 antibody itself. LPS, our positive control, stimulate release of all cytokines to the levels higher than saline, as expected. Control antibody was no different than a saline control. Anti-CD820 antibody increased as compared to saline control, interferon gamma, interferon gamma, interleukin 1, interleukin 2, interleukin 6, interleukin 8, TNF alpha, and granulocytes, monocytes, colony stimulating factor, which we recognize as all major pro-inflammatory cytokines. Therefore, we proceeded with evaluating this plurality of cytokines for their effect on CYP enzymes in co-cultures. These slides illustrate indirect suppression of CYP1A2. Primary cultures of human hepatocytes from free donors were incubated for 72 hours with medium alone, which is here, and here, which is our control, medium containing IL-6, which is our positive control, here for mRNA and here for activity, as well as antibody itself for measurement of a direct effect, and plasma from antibody-stimulated blood for measurements of indirect effects. Our control, interleukin-6, had mild suppressive effects on both message and activity of CYP1A2, as expected. The antibody did not have direct effects, neither on activity nor the message. But cytokines released by antibody in blood suppressed both mRNA and activity of CYP1A2. Similarly to what was observed for CYP1A2, the antibody did not have direct effect on CYP2B6 mRNA or enzyme activity, but it suppressed both the mRNA and the enzyme activity through the pro-inflammatory cytokines, as illustrated here. Indirect suppression of message and indirect suppression of activity. CYP3A4 was the third enzyme evaluated in this study. Interleukin-6, our positive control for suppression by a large molecule, had a strong suppressive effect, as expected in case of CYP3A4, which is very sensitive to interleukin-6. Again, the antibody did not have direct effect on the message or activity, but indirectly suppressed message and activity of CYP3A4. Our conclusions for CD28 case are that LPS and ANC28 antibody cause the release of multiple cytokines from whole blood ex vivo. Plurality of the cytokines suppressed the mRNA and the enzymatic activity of hepatic CYPs in vitro, while the antibody itself did not. Therefore, DDI potential of certain therapeutic proteins, such as ANC28, can be identified in vitro, provided that cytokine release in blood is taken into consideration. I'd like to go through the second case study. The second case study is that of TV1106, albumin-fused human growth hormone. TV1106 is growth hormone genetically fused to albumin. Fusing of growth hormone and albumin, a carrier protein without hormone activity, but with a long plasma half-life, extends systemic circulation of growth hormone and preserves hormone's activity. TV1106 is being developed for treatment of growth hormone deficiency to provide a sustained exposure. The aim of the study was to evaluate whether genetically fusing growth hormone to albumin changes drugs' effects on drug metabolizing enzymes. Such changes could affect DDI potential of the modified peptide. I need to add that growth hormone is known to stimulate cytokine release and to have effects on drug metabolizing enzymes. Focus of the study was on cytokine-mediated suppression of drug metabolism by unmodified and albumin-fused peptide. According to our experimental design, we incubated blood with the controls, which is saline and LPS, as well as unmodified growth hormone and the test article, TV1106. LPS plasma contained higher level of all cytokines tested as compared to saline plasma and as expected. TV1106 plasma had less cytokines than growth hormone plasma, with exception of interleukin-12, which was not statistically significant. Only statistical significant difference was that for pro-inflammatory interleukin-6. In this case, growth hormone resulted in a cytokine concentration or interleukin-6 concentration of 880 picograms per ml versus about 80 in modified growth hormone. We proceed with the evaluation of the effects of this plurality of cytokines on CYP enzymes in hepatocyte co-cultures. In this experiment, the hepatocytes were treated with three concentrations of plasma, 10, 20, and 50%. Interleukin-6, our positive control, suppressed both CYP1A2 mRNA and enzyme activity, as expected and illustrated here. As expected, the LPS plasma strongly suppressed CYP1A2, which is indicated here. LPS plasma, strong suppression of message and activity. TV1106 did not have direct or cytokine-mediated significant effects on CYP1A2 mRNA or activity, direct or indirect. Growth hormone indirectly suppressed both mRNA and activity of CYP1A2 in agreement with what I mentioned to you that this is known to stimulate cytokine release in vivo. The effects of the drug on CYP3A4 can be summarized as follows. Drug did not have a direct effect on the CYP, while positive control suppressed the RNA and enzyme activity. Drug did not have cytokine-mediated effects on the CYP, while growth hormone plasma suppressed CYP3A4, mRNA, and enzyme activity. You see there is no indirect effect on the modified hormone of 3A4. There is effect, indirect, of cytokines released by unmodified hormone. And again, of course, LPS plasma had a strong suppressive effect. In this study, TV1106 did not have direct or cytokine-mediated effects on a third CYP, CYP2C19, mRNA, or enzyme activity. These data are not shown. Our conclusions of TV1106 case are that treatment of blood with growth hormone increased multiple cytokines, while treatment of blood with TV1106 had no effect on the 9 cytokines. Growth hormone itself had little or no direct effect on CYP1A2 or CYP2C19 mRNA, but increased CYP3A4 mRNA twofold. That data was not shown, but the growth hormone indirectly suppressed CYP1A2 and CYP3A4 as shown on the previous slides. TV1106 had little or no direct or indirect effect on CYP's mRNA or activity. Therefore, in contrast to growth hormone, albumin-fused TV1106 was unlikely perpetrator of CYP1A2 to C19 or CYP3A4 direct or cytokine-mediated DDI. Our last case study is that of tilsotolimod. This work was presented at the American College of Toxicology meeting, and the published abstract is a reference here, also available on our website. Molecules discussed in 2 previous cases were proteins or peptides, and tilsotolimod is an oligonucleotide drug. The premise of the study was that, although not a peptide drug, it is possible that immunomodulator, such as oligonucleotide tilsotolimod, an agonist of Toll-like receptor 9, designed to enhance T-cell responses to tumor antigens, could precipitate drug-drug interactions. Tilsotolimod is intended for intratumoral injections. Tilsotolimod alters the tumor microenvironment by improving antigen presentation by dendritic cells and macrophages with subsequent proliferation of antigen-specific cytotoxic T lymphocytes, which are proliferating in the site of injection, as well as the distant tumor cells, resulting in cell death in both locations where drug was injected and other locations in the body. Human trials of the drug are ongoing, and I expect that clinical data describing changes in the cytokines will be available for comparison with the in vitro data. Following our study design, blood was treated with controls, saline and LPS, and 2 concentrations of the drug. I would like to add that LPS is an agonist of toll-like receptor 4, while tocilizumab is an agonist of toll-like receptor number 9. Activation of toll-like receptor 4 and 9 resulted in different profiles of cytokine release, specifically higher levels of interleukin-2, interleukin-6, interleukin-12, and tumor necrosis factor alpha were found in LPS plasma. Tilsolomod plasma contained increased amounts of interferon gamma-induced protein 10, IP10, monocytes chemoattractive protein 1, as well as MIP1 and tumor necrosis factor alpha. Most notably, interferon alpha 2A was increased in tilsotolimod plasma about 1,500 times as compared with LPS-treated plasma. I also would like to know that monocytes chemoattractant protein 1 was also increased in tilsotolimod plasma more than in LPS. Interestingly, I'll add that LPS plasma contained significantly lower amount of this molecule than saline plasma. Effects of the drug on the mRNA are presented on the left-hand panel of this slide, and the effects on the enzyme activity as presented on the right. In this discussion, we call increase over 50% an induction and decrease of over 50% a suppression. We observed that tilsotolimod did not have direct or cytokine-mediated on CYP1A2 mRNA and did not have direct effects on enzyme activity. But the drug, indirectly through the cytokines released to the blood, suppressed CYP1A2 activity by more than 50%. Tilsotolimod also indirectly suppressed CYP2B6 activity. Drug did not have direct or cytokine-mediated effects on mRNA or activity, but it indirectly suppressed CYP2B6 activity by about 80%, more than 80%. In this study, we also evaluated CYP3A4, and CYP3A4 was not affected by drug either in a direct or cytokine-mediated level, therefore the data are not shown. Our conclusions for tilsotolimod case studies are that agonists of toll-like receptors 4 and 9 produce different cytokine stimulation profiles. Tilsotolimod suppressed CYP1A2 and CYP2B6 enzyme activities in cytokine-mediated way. Drug did not have direct effects on CYP1A2, 2B6, or 3A4. Therefore, our conclusions combined from 3 cases we presented are that cytokine release by PBMCs, Kupffer cells, and others should be accounted for evaluation of DDI potential of therapeutic proteins, and Sekisui XenoTech method incorporates the effects of these molecules in its design. Presented cases have demonstrated utility of the in vitro study design. I'd like to also indicate that current regulatory guidance is minimal at the moment. As we know, FDA just solicited comments for a new guidance document, and therefore I think that as more studies conducted according to this study design are made public, it will be easier to formulate recommendations for the use of the assay as a predictive in vitro tool. More specifically, which immunomodulators can be meaningfully evaluated in the assay, and what cytokines, at what level of their stimulation in vitro, warrant moving past the cytokine release assay into the stage of treating hepatocytes co-cultures with the plasma. At that moment, I would like to thank you for your attention and look forward to answer your questions, either now or later by email. If time is not permitting, I will answer all your questions directly by email.
Thank you, Maciej. While we wait for questions to come in, I wanted to mention that information about our services, papers, posters, other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Maciej's presentation or any of Sekisui XenoTech's contract research services or products, please feel free to ask them in the post-webinar survey or contact us through our website.